Hello, everyone, and welcome to the Sugar Science Ask the Expert. I'm Alexis Carmer, and I'm filling in for Monica today. The title of today's presentation is Interferons are the key cytokines acting on pancreatic islets in type 1 diabetes. It's my pleasure to introduce our speaker, Dr. Alexandra Coomans De Brachene. Dr. Coomans De Brachene is a postdoctoral researcher at the ULB Center for Diabetes Research in Brussels. She obtained her PhD in biomedical and pharmaceutical sciences at UCL in Belgium, and then joined Professor Decio Ezrich's group to pursue her career on the pathogenesis of type 1 diabetes. She focuses on the study of innate immune and antiviral responses in human beta cells to try to understand what triggers the innate immune response in the early stages of the disease and to identify relevant targets for therapy. Alexandra, we're really looking forward to your presentation. Thank you for sharing your work today. Just so everyone knows, there will be time following the presentation for questions, which you can unmute yourself for at that time or put in the chat box. I'll hand it over to you now, Alexandra. Thank you.
Hello, everyone. Thank you very much for the introduction. And thank you for the invitation. So I'm really happy to present you some data that we have generated in the lab and that we have recently submitted for publication. So you can already access the full paper in bioRxiv if you are interested in. So the title of the presentation is Interferons are the key cytokines acting on pancreatic islets in type 1 diabetes. And before I started to present the study, I just want to mention two of my colleagues who did this work with me. So Maria Inés, who was a postdoc in the lab and she left a few months ago, and Florian, who is finishing his PhD for the moment. And they did all the bioinformatics analysis of the study. So they did really an amazing job. And this study was done under the supervision of Decio Ezrich. We also had a lot of collaborators, as you can see here, so many members of our group at the UCDR, but also people from the University of Exeter in UK, in Pisa, Italy, in Lille, France, in Barcelona, Spain, and also from the company Interferons in Switzerland. So thank you to all these collaborators. So type 1 diabetes, well, you all know the disease, so I will not make a big introduction. I just want to remind you the dialogue that exists between the beta cells and the immune system, and that is really important in type 1 diabetes. So when the cells are exposed to a dangerous signal, such as viruses or an endogenous ligand, there is a recognition by pattern recognition receptor, and it leads to the activation of the innate immune response and the production of type 1 interferons, so interferon alpha and beta. So these cytokines, they can bind on their receptor to activate the cytokine receptor signaling. And it will lead to an increase in MHC class 1 at the cell surface, you can see here. There will be alternative splicing, ER stress, and this can lead to the increase in presentation of modified antigens. There is also more apoptosis, an increase in checkpoint proteins such as PD-L1, and importantly, the expression of chemokines and cytokines. And so these chemokines and cytokines, they will attract and activate different immune cells that will in turn start to secrete cytokines and chemokines. And so these cytokines, they will bind on the receptor of the beta cells, and they will further increase MHC class 1 expression, presentation of antigens to recruit more immune cells and amplify insulitis. So during insulitis, pro-inflammatory cytokines are released by the immune cells, and they can act on beta cells to maintain and to amplify this dialogue between the beta cells and the immune cells. And so this creates a vicious circle. So pro-inflammatory cytokines really play a central role in type 1 diabetes by controlling innate and adaptive immune responses. And so they impact on the initiation and the amplification of beta cell autoimmunity. And so the importance of cytokines in human type 1 diabetes are evident by different clinical data. So interferon alpha, for example, is expressed in islets of patients with type 1 diabetes. And there is a type 1 interferon signature in individuals at risk of developing type 1 diabetes. And this already before the development of autoantibodies. There is also an interferon gamma signature in islets and sera from individuals affected by type 1 diabetes. Elevated levels of TNF-alpha are found in the plasma and the islets of patients with type 1 diabetes. And TNF-alpha levels are associated with an aggressive phenotype of early onset type 1 diabetes. And serum levels of interleukin-1 beta are also higher in type 1 diabetes patients when compared to non-diabetic individuals. So based on this evidence, the impact of cytokines on human pancreatic beta cells is largely studied. And so usually what we do is to use cocktails of 2 to 4 pro-inflammatory cytokines and expose pancreatic beta cells to this mix. And when we do that, we can see inflammation, HLA class 1 expression, ER stress, and apoptosis. And these are all hallmarks of type 1 diabetes. But when we do this, we don't know which cytokine or cytokines are the main drivers of these effects. And of course, if we want to identify relevant therapeutic targets to preserve the beta cell mass, it's really crucial to identify the specific role of each individual cytokine on pancreatic beta cells. And so this is the purpose of the present study. So to do this, we use the human beta cell line endog beta H1, and we expose these cells to interferon alpha, interferon gamma, interleukin-1 beta, or TNF-alpha during 24 hours, and then we performed a deep RNA sequencing analysis. With this volcano plot, you can already see in red the genes that are significantly down or upregulated by the different cytokines. And so just looking at this, you can already see that the cytokines have a distinct impact on gene expression. So when we look in this table, the number of genes that are up and down regulated, you can see for interferon alpha and interferon gamma that there are several thousands of genes that are significantly regulated. For interleukin-1 beta, it's only just a bit more than 1,000. And for TNF-alpha, less than 100 genes were affected. So we were really surprised by this data, especially for TNF-alpha. And so we checked the expression of the cytokine receptors in different beta cell models from RNA-seq data. So in endog beta H1 cells, human islets, FACS-purified beta cells from control individuals, and from type 1 diabetes individuals. So you have the TNF receptor, interleukin-1 receptors, interferon gamma, and interferon alpha. So you can see that all receptors are expressed in the different models, and there is a predominance for the interferon receptors. And here I just have to mention that there is a huge difference with the rat beta cells, where you can see that the interleukin-1 receptor is much more expressed than the others. And so this can explain also the difference that we can see regarding the impact of the different cytokines between the rat and the human. So this is really important. So then we wanted to see which pathways are regulated by the genes that are modified, and we did a gene set enrichment analysis. So here I presented the top 20 up and down regulated pathways with the different cytokines, but this is of course really small, so I highlighted some important pathways. So you can see for interferon alpha and interferon gamma, it's mainly interferon signaling related pathways, antigen processing and presentation, RIC1-like receptor, antiviral response. We have also here the assembly and peptide loading of HLA class 1, so really an immune response. For interleukin-1 beta, we have translation regulation, nonsense mediated decay, also here, activation of NF-kappa B interleukin-1 signaling, and for TNF-alpha, eukaryotic translation initiation, RNA processing related pathways. So it's really clear with this first analysis that interferon alpha and interferon gamma induce a more important immune-related response in human beta cells than interleukin-1 beta and TNF-alpha. So we wanted to confirm this in vitro by investigating the regulation of some immune-related genes relevant for the disease. So we selected PDL1, HLA-E, HLA-ABC, and CXCL10. because the protein coded by these different genes is highly expressed in islets and in serum of individuals with type 1 diabetes. So this is really relevant for the disease. So we exposed end-of beta H1 cells or human islets to the different pro-inflammatory cytokines. And you can clearly see that only interferon alpha and interferon gamma induces the expression of the 4 different genes in the 2 different models, while interleukin-1 beta and TNF alpha had no impact. Then we wanted to compare the genetic signature induced by the different cytokines to the genetic signature present in fact purified beta cells from type 1 diabetes patients. So for this, we did a rank-rank hypergeometric overlap analysis. So in this analysis, you can compare 2 ranked lists and see how much similarities you can find between these 2 lists. So to make it simple, for example, here, you can see the genes that are downregulated or upregulated by interferon alpha versus control, and to compare these to the genes that are downregulated or upregulated in beta cells from type 1 diabetes individuals versus control. And so in this area, you can see the genes that are commonly upregulated between the 2 lists. Here, the ones that are commonly downregulated, and here those that are regulated in the opposite direction. So we did this analysis for the 4 different cytokines compared to type 1 diabetes. So we first observed that there was a predominance of commonly upregulated genes between the different pro-inflammatory cytokines and type 1 diabetes samples. And also there is a closer correlation between interferons, so interferon alpha and gamma, and type 1 diabetes samples. And so we wanted to see what are the regulatory pathways in the commonly upregulated genes. So we did this analysis for all the cytokines, so checking this area. So for interferon alpha, we can find interferon signaling mainly, antigen processing, antigen presentation, chemokines. For interferon gamma, it's more or less the same, so interferon signaling, MHC class 1 mediated antigen processing presentation, antigen presentation, antiviral mechanism, chemokines, etc. So again, really an immune response. By contrast, for interleukin-1 beta and TNF alpha, the commonly upregulated genes with type 1 diabetes were more related to extracellular matrix organization, histone deacetylation, histone acetylation, DNA methylation, etc. So again, this analysis confirms that interferon alpha and interferon gamma are the main drivers of the immune response in human beta cells in the context of type 1 diabetes. And so to really see the importance of interferon in beta cells from individuals with type 1 diabetes, we looked for an interferon-stimulated gene signature in beta cells. So for this, we analyzed a single-cell RNA-seq data from the HPAP. So there we had access to beta cells from non-diabetic control, so the ND, from individuals presenting 1 or 2 islet-specific O2 antibodies, here and here, and type 1 diabetes donors. And so these were analyzed by a single-cell RNA-seq. So for each cell, we determine an ISG score. And so each dot represents a cell with this score. And you can see that there was an increase in the ISG score that positively correlated with the natural evolution of the disease. So you can see a lower score in the non-diabetic subject, and then a progressive increase and the higher score in type 1 diabetes beta cells. So this again confirms the key role and really the relevance for interferons on human type 1 diabetes. Then we decided to study the regulation of different groups of genes that are relevant for the disease. And we started by the regulation of genes whose protein is highly expressed in pancreatic islets from type 1 diabetes donors when compared to controls. So we selected 12 genes, and this selection was based on their high protein expression in islets from type 1 diabetes individuals from the present and previous studies. So for example, here in the present study, we have shown that HLA-G is expressed in islets from type 1 diabetes. So you can see this in this pancreas tissue section, where you have the non-diabetic individual. So you can see the staining for insulin, there is no HLA-G. In type 1 diabetes, insulin deficient islets, so you can see the islets with the glucagon, but there is no insulin and no HLA-G. And in type 1 diabetes, insulin containing islets, so you see the insulin and there is a staining for HLA-G. So this is the list of the 12 genes, so you can see here HLA-G. So the way we presented it, you can see the different colors and the values. It represents the log2 fold change, so you have the scale here. And we put it only the genes that are significantly regulated. So that's why there are some empty spaces when the gene was not significantly regulated in this condition. So first you can see that both interferon alpha here and interferon gamma here induced these 12 genes and with a really high fold change. Then interleukin-1 beta and TNF-alpha, they had only a mild impact. So you can see really few genes induced and a really low fold change. Then we looked also at the regulation of type 1 diabetes candidate genes. So more or less 80% of the candidate genes are expressed in the beta cells. For example, TYK2 downstream of interferon alpha receptor, MDA5 for the recognition of viral infection, PTPN2, BAC2, etc. So here we have a list of 66 candidate genes that are expressed in beta cells and that were regulated by at least one of the cytokines. So the representation is the same as the previous one. You can see that most candidate genes are regulated by interferon alpha and by interferon gamma, and only few by interleukin-1 beta and TNF-alpha, only one. So then we looked at the regulation of genes that are related to antiviral and inflammatory pathways. So in pancreatic beta cells of type 1 diabetes individuals, there is a strong antiviral and inflammatory gene signature. So we wanted to investigate the impact of the different cytokines on the expression of some genes related to these pathways. So here we selected 17 genes that you can see here. Most of them were modified by interferon alpha and by interferon gamma. Interleukin-1 beta had a limited impact, and TNF-alpha did not affect the expression of these genes, so that's why it's not represented. And so from this list, we were interested in this gene, GNFX1, because it was recently identified as a double-stranded RNA sensor important for the early antiviral response. So just to show you why it could be important, so I come back to this picture. I told you that viruses can initiate the innate immune response. So when the beta cell is infected by a virus, such as the Coxsackie virus B, the double-stranded RNA of the virus is recognized by the double-stranded RNA sensors, RIC1 and MDA5. So these two are well described. And it leads to the innate immune response with the production of type 1 interferon that can bind on its receptor and initiate the dialogue with the immune cells. So we were wondering if the GNFX1 could also play a role in this process. So first, we wanted to see if it was expressed in beta cells of individuals with type 1 diabetes to see if it is really relevant to study this gene. So again, we analyzed the single-cell RNA-seq data from HPAP. And this is really interesting. You can see a gradual increase in the percentage of beta cells that express GNFX1, as you can see with the size of the circle, and also an increase in the average expression in beta cells, so as you can see with the different colors. So going from the non-diabetic to the autoantibody positive individuals and type 1 diabetes donors. And we also see this progression for MDA5 and RIC1. So based on this data, it seems that GNFX1 regulation is really relevant for type 1 diabetes. And so first, we confirmed the regulation by interferon alpha. So in endog beta H1 cells, you can see at the protein level a nice increase after 8 and 24 hours with the quantification here. And at the mRNA level, already after 2 hours of stimulation. We also looked for the mechanism, so you can see that interferon alpha induces the expression, and this is completely abolished with the JAK inhibitor baricitinib. The induction by interferon alpha is also significantly reduced by the silencing of STAT2, but not STAT1. And here in human islets, you can also see the nice induction of GNFX1 by interferon alpha which is prevented by the TYK2 inhibitor BMS. We also wanted to see if ZNFX1 could be induced by a viral infection. So here we used the double-stranded RNA analog poly-IC, or PIC, which is used to mimic a viral infection. So again, you can see that poly-IC strongly induced ZNFX1 expression at the protein level here and here at the mRNA level. So with this data, we wanted to investigate further the role of ZNFX1 in the antiviral response in human beta cells. So I put this picture here to help you to follow what I wanted to do. So I was wondering if I silence the expression of ZNFX1 and I expose the beta cells to poly-IC, are the cells still able to induce the innate immune response with the production of type 1 interferon and all the downstream signaling leading to the production of ISG? So here you have the expression of ZNFX1. You can see that it is well induced by poly-IC, and we have a nice repression with the siRNA. So first, I looked at the production of type 1 interferon. So interferon beta mRNA, and here the protein released in the supernatant, so by ELISA we measure. We can see a good increase of interferon beta with poly-IC, but this effect is strongly reduced when ZNFX1 is silenced. Next, we also looked at the phosphorylation of STAT1 and STAT2 downstream of interferon alpha receptors. Poly-IC induces strongly the phosphorylation of STAT1 and STAT2. And again, we have a strong reduction when ZNFX1 is silenced. And here you have the quantification. And finally, we looked at the expression of different ISG. So the double-stranded RNA sensors, RIC1 and MDA5, the chemokines CXCL10, and here at the protein level in the supernatant, the antiviral gene MX1 and HLA-ABC. So they are all induced by poly-IC, and you can see a strong reduction when ZNFX1 is silenced. And these data, they identified ZNFX1 as a novel important player of the antiviral response in human beta cells. So to conclude, we observed that interferon alpha and interferon gamma are the two main pro-inflammatory cytokines responsible for the transcriptomic modulation of beta cells in the context of type 1 diabetes. They induce a gene signature that closely correlates with the gene signature observed in beta cells from individuals with type 1 diabetes. Immune-related genes such as HLA-ABC, HLA-E, PD-L1, and CXCL10, which are relevant for the disease, are induced in human beta cells by interferons, but not by interleukin-1 beta or TNF-alpha. The double-stranded RNA sensor ZNFX1 seems to be a critical regulator of the interferon-mediated defense responses in human beta cells, and it could be an important player in the initiation of the innate immune response in the early stages of type 1 diabetes. So finally, therapies that aim to protect beta cells from an immune attack should focus on interferon alpha and interferon gamma to stop the dialogue between beta cells and immune cells. And so with this, I would like to thank all the members of my team at the UCDR, and thank you for your attention.
Thank you, Alexandra. That was a very interesting presentation.
Thank you.
Now I'd like to open the floor to questions. If you have a question, please unmute yourself or write it in the chat box if you prefer.
Alexandra, perhaps I can start with a question that you already discussed. So when you knock it down ZNFX1, you didn't see a complete inhibition of interferon beta or of other downstream genes. Do you think that this is because the inhibition was just partial, or you have other signaling going on?
So yeah, this is a point that we already discussed because there are different possibilities. So indeed, the silencing of ZNFX1 is not complete, so it remains some expression. So what I did is to see if there was a correlation between the silencing of ZNFX1 and the decrease in gene expression. And actually, we found a really nice correlation. The more important was the silencing, the more important was the decrease in genes. So this is really interesting. But another possibility is a compensation by MDA5, because ZNFX1 and MDA5, they both recognize long double-stranded RNA. So maybe by reducing the expression of ZNFX1, MDA5 can compensate and take the role of ZNFX1. So this is ongoing for the moment, so I cannot really comment on this. I'm doing a double silencing to see if we can reduce more, but this is really preliminary. So for the moment, I don't have an idea about this.
Alexandra, I have a quick question. You had a slide that showed the signature, the gamma signature in the control group, and then the groups with 1 and 2 antibodies and the T1D group. And I don't know if those T1D subjects were recent onset. And I was just wondering, do we know anything about how this changes with longer duration, that signature with longer duration of T1D?
That's a really good question. I don't know actually if they were recent onset or not. Maybe I see Xiaoyang is there and she did the analysis. I don't know, Xiaoyang, if you know for how long they had the disease, because she did this analysis. So I don't know if this is an available data or not.
Sorry, it's not in my brain, but I can check. I have this data.
Okay.
I don't remember the duration.
Do you mean, I know this, it's for the bulk RNA seq or for the single cell?
This is the single cell where you have the non-diabetic, the autoantibody positive, and the type 1 diabetes.
Yeah, you have different durations of disease, but I think a very interesting group in this context is the double antibody positive individuals, because these are the people before the disease started. So perhaps, Alexandra, you can comment, you know, how they behave regarding antiviral responses.
Well, maybe I can just put it back so everyone knows. Well, there is this one for the ZNFX1, but maybe you were more mentioning the one for the ISG score. But well, they progress the same way. So you really see from the non-diabetic, and so it's the same for the ISG score, that it's really low. And then you have this increase with the autoantibody positive with only one autoantibody. So there is a low risk to develop the disease. And when they have 2 autoantibodies, well, minimum 2 autoantibodies, the risk to develop type 1 diabetes becomes really high. And you see then the increase in this ISG signature, and then it's even higher when the people develop type 1 diabetes. So you really see the correlation, the progression in this ISG score with the progression of the disease.
Just to follow on that, you had mentioned early in the talk about the known marker in the sera that's seen before the autoantibodies, the interferon signature that's seen in the at-risk individuals before autoantibodies. Could you just comment on, you know, if we're pursuing this type of therapeutic, potential biomarkers and the ability to follow the treatment with a biomarker?
Well, it's a complicated point. Well, it's true that so interferon alpha is present really early. So there is already something happening before we can detect the autoantibodies. So if we take the autoantibodies as a signal or as a biomarker that the disease is ongoing, then maybe it's already too late. Well, not too late, but it's already after the interferon alpha, and we could start treating the patients before this. Well, I think then to detect a type 1 interferon signature, or the presence of interferon alpha in individuals before type 1 diabetes, before the presence of autoantibodies, in the general population, it seems difficult. Maybe, again, people that are at risk and that we want to follow as early as possible, try to detect it in the serum. but I don't know if it is feasible.
It's too dispersed. You can have statistical significance, but you cannot use this as biomarker. Just there is a question from Young Dai. Please take a look in the chat. Can you see it?
Yes, it says, do beta cells produce interferons?
Yes, but not a lot. So when we expose the beta cells to poly-IC, they produce interferon beta. So this is what I showed. It doesn't move anymore. So here you have interferon beta released in the supernatant of the cells, but this is not a huge induction. And so it's mainly the other cells in the environment that will produce it. So there is some secretion, but not a lot.
I had one other question, just that I wrote down after I looked at your paper, and I was curious your thoughts about, we're talking about the possibility of using combination therapies, and do you think that blocking an individual cytokine will be enough given the redundancy in that pathway?
Yes, I was thinking also about these points. Of course, well, we don't know. For the moment, for example, blocking, if you try to block only the immune system with anti-CD3 molecules, it gives really nice effect. It delays the disease by 2 years, but it's not sufficient alone. So maybe if we target one cytokine to block the immune attack on the beta cells, it will not be sufficient also. But so it's not excluded that the combination will be the best. But what we think is that if we target interferon alpha or interferon gamma, based on the data that we have here and also on different studies, we can reduce MHC class 1, we can reduce the production of chemokines, so we can reduce the attraction of the immune system to the beta cells, and so it can stop this dialogue, because if the immune cells are not recruited anymore, they will not secrete the other cytokines, interferon gamma, interleukin-1 beta, and TNF-alpha that are produced only by the immune cells infiltrating the islets. So if we can stop this dialogue, then maybe just inhibiting these cytokines can be beneficial.
Okay, well, thank you so much, Alexandra, for the beautiful presentation and for this discussion from the participants. There was a lot of interest in your talk, and it'll be posted on YouTube shortly for other people that are interested that weren't able to join today.
Okay, thank you very much. Thank you for the invitation.
Will lead to further conversation and collaboration. Thank you.
Great.
Thank you.
Thank you.
Thank you, everyone.
Bye.
Bye.